Okay, so welcome participants again to this CSIR JIGYASA webinar series. Let's talk science, what, why, and how. So for today's talk, we have with us Dr. Mitali Mukerji for her lecture on genomics, connecting sciences. So I would like to take this opportunity to introduce her first. Dr. Mitali Mukerji is a chief scientist and heads the Genomics and Molecular Medicine Department at CSIR Institute of Genomics and Integrative Biology. She has a PhD in molecular genetics from Indian Institute of Science, Bangalore. She is currently heading Genomics Group in the Center of Excellence on Applied Development in Ayurveda, Prakriti and Genomics, supported by Ministry of Ayush. She has been involved in developing a new area of research, Genomics, which aims to integrate Ayurveda with modern medicines through genomics. Her group has provided the first evidence for molecular correlates of Prakriti that lead to identification of a predictive biomarker for high altitude adaptation. She is also recipient of several prestigious awards, including CSR Young Scientist Award, DBT Young Women Scientist Award, Shanti Swarup Bhatnagar Award, Vasvik Award, and is also Fellow of Indian Academy of Science. So with this brief introduction, I welcome you, ma'am. And now I request you to deliver your lecture today for the students and teachers who are waiting to hear you. Thank you.
Thank you so much. Thank you for giving me an opportunity to interact with the students here. And I will try to kind of excite the audience about the different facets of genomics through the lecture. It will be primarily more of an introductory kind of a lecture, not very specifics about the kind of work we are doing, except for some brief descriptions. And then we will have some interactions with the students. So I'll start my. So is it fine? The slides are visible. Hello, Pooja.
Ma'am, no, yes, your screen is not shared. Could you please share the screen with us?
Okay, I just thought I shared the screen. Okay, let me see. Share screen. Is it coming on? Hello, is this coming on?
Not yet. Not yet. No.
I'll.
Camera and then share the screen. I think
I shared. Maybe I'll just do it again.
yeah. Please. Please share the screen. So some technical error is coming. Yeah.
Just one. Is it okay?
Yes, perfect. Yes,
Okay.
ma'am. Please.
Okay. So sorry for that small technical glitch. So today what we are going to talk about is how do we connect different sciences through genomics. So just prelude to that, I have a certain set of questions where which most of us one time or the other would have asked. That questions like what makes us human? How are we different from the other animal kingdom? Why do we look so different, maybe in terms of skin color? When we talk about that we have descended from apes, do we really know that they were our ancestors? Or even for that matter, if we say 100,000 years back we came from Africa, all of us have come out from Africa, then how do you know that? There are other kinds of things. As school kids, we sometimes are blamed for being lazy or getting up late. So do you think you need to be blamed or your genes need to be blamed? All the microbes that are around us, do we meaning are they important to us or they are always harmful? Or questions like when did humans start wearing clothes? Because we know that early apes did not, or early humans, we do not know. We don't have an evidence as to which time people would have started wearing clothes. The other kinds of questions which also people like ecologists ask is like, can we count the animals in the forest without seeing them? Or how do bats learn to ecolocate? Meaning, what is the mechanism? Now, when we are talking about corona, then why don't bat get infection? And if they have infection, do we need to worry about it? The other kinds of questions in plant kingdoms of we can ask why is there so much water in watermelon? It grows in deserts, but still it has been able to conserve so much of water in them. Or there are animals which live in lots of extreme environments. What has given them an advantage to survive in such kind of environment? For example, heat, cold, extremes of cold. Everywhere you go, there are some amount of animals or bacteria. They are surviving in that environment. Or questions like how do honeybees have differences within themselves? That somebody is queen, somebody is drone, somebody is worker. Everything comes from the same kind of a genetic makeup, but how do they become different? And the other kinds of questions are, can we understand things from the past? For example, why populations became extinct? Or were there some things like epidemic in the earlier times, and how did people deal with it? Can we get some kind of questions from looking at history? Currently, even if we look at very many people's families sometimes suffer from diseases, so how do we get some kind of cure across to them? Then there are more pertinent problems. Nowadays, we are totally dependent on digital information. We have images, we have lots of information all getting collected all over the world in the cloud and all. How do we will surely run out of storage space. So can we actually look at it? And then going back to the other kinds of things, which is totally the other way around, that we have an ancient system of medicine called Ayurveda. And some people think that it's a mythology. Can we actually find ways to test this out? So these are some of the questions I've just listed, but you might have more curiosity questions. And what I'm trying to would try to tell you in the next half an hour or so is that, believe it or not, many of these questions can be addressed to some extent by this area of science, which is called as genomics. And I want to excite all of you to this interesting science. So before that, I am kind of flashing some lines over here. As you can see, most of you might not be able to read all the lines, right? Because they are in different languages, right? But if we do a translation of all these languages, then we might see that every line is telling the same thing. It is, we can do it. But why we could not read all these lines? That is because it was all written in different languages, and we cannot master all these languages, right? But we are lucky as human beings that for us, all the animal kingdom, nearly all the animal kingdom, be it bacteria, be it mouse, be it chimpanzee, gorilla, humans, plants, all of them, when they have to express themselves, or when they have to kind of all whatever they are all driven by four chemical letters, meaning so every plant, every human, everybody shares the same set of four chemical letters, which was elucidated by you might know by Watson and Crick way back in 1950s. And that gives us an advantage that we can understand every living organisms by these four letters. So what are these four letters called? These four letters are called as nucleic acid, and when they get threaded together, so they are like the alphabets. When they get threaded together, they become words, which are called as DNA. And you have a thread of DNA, it makes the genes, and you stream them together, the sentences like you have in a book, to a chapter. Then this becomes a chromosome, and all the chromosomes, if they are compiled in one place, that's called as the book of life. Just like chapters can be merged together to form a book, and that book is called as a genome. So we have around 3 into 10 to the power 9 such letters inherited from each of our parents, and these are called as the genomes. And the science of understanding what these letters mean in terms of how we are, what we are, is what is called as the science of genomics. So, just to kind of get back to some of the textbook stuff, like we all know that there are cells in the body. There are around 10 to the power 14 cells in the human body, and each cell would have all these 23 pairs of chromosomes. So, the entire genome is there in all these cells, right? But then there are different organs. For example, there is stomach, there is blood, there is nervous tissue. So what happens there? It is not that we lose some of our genetic material, only some part of the genetic material open up to kind of say, okay, now you make stomach, or now you make a nervous tissue, now you make a blood. But all the cells in our body have all these genetic material in them. The second question is that, is all of it same between you, your sister, your parents? Clearly, if you are inheriting two sets, then they would be different. But what also happens is that as these book of life gets written across generation, then sometimes some changes can creep in, just like as you are having a word like color, and then you can spell it a little differently. Sometimes it might have a meaning because different countries use different kinds of this. But then if it changes to another one for by an insertion in the word, then it does not make any sense, and it might get lost out unless it again actually changes to something which has a meaning. So for example, from this word called cooler or whatever, if it becomes cooler, it still is not color, but it has a change. And then you could have deletion, where the words can get broken. Then that can also be spelled a little differently, like in the SMS language of today's, many of the spellings you keep on saying differently. Then it can get inverted, so you can actually have a cool can become look, and then there could be fusion so that you actually have another word called cool look. So you started with color, and you had various kinds of roots that word can take, and as a result of which sometimes it can totally change. So this is what has happened across millions of years. We have this book, but this book has undergone different kinds of changes because people moved to different parts of the country, parts of the world, and as a result of which there are various books of life, the same kind of book, but a little different sometime in nature. Now, what happens to this book of life? So this is the genomic part, which is the 4 letters, and now it has to make meaning out of it. So what it does, it actually converts or transcribes itself to something called a messenger, and that messenger has a message that what protein needs to be made in which cell or which tissue. So the DNA gets converted into a messenger RNA, and then that messenger RNA, three letters are read by the machinery of the cell to understand, okay, if this letter is there, then this amino acid is there. And by that way, if you read these letters, you string the amino acid in a chain by a process which is called as translation, and you get these functional parts of the cell which are called as proteins. So you see, if there is a change in this, then the message might be a little different. And if the message is a little different, then it might end up incorporating a different amino acid because it recognizes this message to incorporate an amino acid. And that can lead to certain kinds of sometimes small changes. Now, some of these changes can become very drastic. For example, you people might have heard about this disease called sickle cell anemia. There is a RBC, there are these genes called beta globin gene. And if there is a small change so that the amino acid becomes different, then you have a drastic change in the shape of this RBC. So, what happens? From the spherical, it becomes this kind of a shape, and as a result of which the oxygen carrying capacity gets a little compromised. So people who would have this change have this condition called sickle cell anemia. So now, once people got to know that there is a genetic material, if there is a change, then such a thing happens. then the idea was that can we read the entire book of life? And if we were to read the entire book of life, then we would have a chance to start comparing it between people. And as a result of which, we might be able to know why people are different, people have different diseases, and lots of other interesting things which I'll be coming to. So that was what was called as the Human Genome Project. It was a very big project which was started in the late 80s, and it took around 13 years for the first draft of the human genome. That is, how what are the core letters in the human genome to be understood. But subsequently, lots of other efforts happened, lots of technological advancements happened, and as a result of which, after the first project, now we have more we have used the same technologies, and we have huge number of human genome sequences available to us. And not only humans, people have used the same technologies to kind of look at the genome sequences of different animal and plant kingdoms, right? And how has this happened? This has happened because of advancements in lots of technologies. Earlier sequencers could not sequence that much length of DNA. It was very expensive. There were the it was very slow. But then over a period of time, there has been rapid advancements in different kinds of technologies. So something which was called as the first generation sequencer evolved into a second generation sequencer. Then it became a third generation. So now the instruments have become smaller in size, and they can actually sequence a large amount of human genome. And what took 13 years, now in 2 days, one day, you can actually read a person's genome fully. So that has revolutionized our capability to read the letters in a very fast space in a large number of people. Now, why do you want to do that? Why? What happens when you get so much of DNA sequences? So the first thing, when the human genome sequence came, we thought that we are humans, we are very evolved. But what came out as some kind of paradox? Earlier, people knew that it is not that humans are more evolved because they have more number of chromosomes. Even a small grass has more number of chromosomes. So, so then people thought that okay, maybe it might be the amount of DNA we have. We find that an amoeba has more amount of genome than human. So that also created a paradox which was called a c-value paradox. Then the thing came that if we sequence the human genome, we would finally know that maybe we have more number of proteins because of which we are so complex. But that was also a dampener, because we got to know that humans also approximately have 20 to 25,000 genes, and so also a small fruit fly or a small grass. So now the question is that even if we have the genome sequence, how do we understand what makes us human and what makes other organisms also equally evolved? So as people are sequencing human genome, lots of stories we can uncover from these sequences. For example, we take the example of python. It can feed on an entire animal, and it can digest it. Any normal human being cannot actually do that. So what is so interesting? And if you look at a python, its organs, it is very small, but then how does it engulf such a huge animal, and how can it digest it without the food wasting inside it, right? So when you go to the genome sequence, you actually find that there are certain kinds of genes which gives it this evolved ability to digest the food, and that is because of its mitochondrial activity, which can do metabolism and generate a lot of heat so that the food can get digested. There are other kinds of things, like there is this very interesting animal called naked mole rat. It lives for a very long time. It lives for 28 years. And what people have found is it does not get cancer. It does not have any pain. So people are now trying to study them. What is there in their genome because of which it actually has that? Or you can think of an oyster which lives in the sea, and it does not have any capacity to kind of feed on what it is feeding. Now, if people are polluting the water bodies so much, then how do oysters still survive? So people have sequenced the oyster genome, and they could get some interesting answers that what makes them survive. Maybe they have a lot of genes which respond to stresses, as a result of which the genes can function better under such harsh conditions. So also people have found that why the queen is a little different, because they make some royal jelly protein which is very unique to the queen bee. Or there are some very very small genomes. One of them is called as urticularia. So how does that survive? So people found that it has got rid of lots of genes which it can do without, and it can actually get all its carbon and nitrogen and phosphorus requirement by just feeding on insects. Or there are these small bacteria which are very resilient to radioactivity. So people found that if you shine or if you expose them a lot to radioactivity, their DNA does not get damaged, they repair themselves very fast. And these kinds of information actually leads us to very interesting ideas about if in nature some evolved function has come out in some animals, do those evolved functions also matter to human, right? So, and then there are other unique questions to humans, because there are some features which are only there in human. That's the brain volume, ability to speak, how we have become bipedal. So when we became bipedal, what are the kinds of changes we've had to have, because all the gravity and all shifted differently. So those are the things. And now if the gene numbers have not changed, what is changing? So it is turning out that lots of repeat, which was thought as junk DNA, is gaining function. People are now trying to understand genomes and trying to see whether repetitive sequences which have expanded during evolution, do they contribute. There are other kinds of things. You might have heard about Neanderthals, which predated humans and had moved out of Africa and were already there till when humans started moving. And people are now finding from fossils, because the DNA is very very stable, so one can actually isolate DNA from fossils or excavations at various places, and then one can sequence them, and then they can compare the sequences with humans. And it turns out that some of the genome of the Neanderthals have are also there in the humans. So it is now being thought that maybe humans came out of Africa, but there were already Neanderthals existing, and there might have been a period in which they might have been intermixing, so that what led Neanderthal to adapt to harsh conditions, those could have been ported into the human genome. So now everybody has 1 to 5% of the genome, which is similar to our very, very, very ancient ancestors like the Neanderthal Denisovan. There are other very interesting things coming up. Earlier, we thought that humans are only made out of human cells, but now we are finding that in the human, there are other kinds of bacteria, microbiome, which also colonize our whole body. So there are nearly a magnitude 10 times more number of bacterial cells in human in different parts of the body. Okay, and these bacteria also come with their own genetic makeup. So these genes contribute to further genes in the human. So now people are studying as to whether these microbes make us human, because very early during the first 1 month, all these microbes colonize all parts of our body, and different people have in the same positions in the body similar kind of microbes. So now people are trying to understand whether these microbes, which symbiotically live in humans, could they contribute. And many striking answers are coming up that the fact that we might be lean or we might be thin is because of different kinds of microbes residing in us. So that has given rise to another area of genomics research, which is called as the Human Microbiome Project. And the purpose is, can we sequence all the genomes in our body? So now there are other kinds of areas. So people are sequencing different kinds of animals like the naked mole rat, the elephant, and all, and trying to compare them with the humans and trying to see whether different kinds of changes which happen in different animals also are shared with the humans. So there are these new projects which are very interesting. You can go to the internet and search for it. There's a project called Earth Biogenome Project, where people want to sequence nearly 1.5 million all species in the Earth. There is a Darwin Tree of Life project by UK, where they want to kind of sequence all the species of animals in the UK. Then bird lovers can understand why different birds are different, because there is a B10K where they are sequenced 10,000 birds to know why different birds behave differently, mate differently, look different, etc. Now, what are the other kinds of things you can get once you know the genome sequence? So there is an interesting area called the museomics. So there's this person called Stephen Schuster. What he does is he sequences different kinds of museum specimens of animals which have gone extinct, like the mammoth, the Tasmanian wolf, and then tries to ask a question: why did the animals become extinct? Are there certain kind of information in the genome? And if we understand that, can we use that information to conserve animals which are in the verge of extinction? And not only he has done this kind of an effort to understand lots of extinct genomes, but he actually used this information to save some Californian condors whose numbers had dwindled to 40, and he could find certain kinds of interesting insights into the genome as to when the animals are going it's extinct, what happens to their genome, so that you could mate the animals in a captive setting to be able to then bring them back from the verge of extinction. So these are some interesting areas. Then there are these interesting things, like I said, humans have microbes in them, so also the leeches, so also the animals which feed on other animals in the forest. So what one can do is actually take the blood out of the leech and sequence that entire blood, because it will have signatures of what animals it ate. And like that, people are now prospecting in the jungles using these kinds of sequence from the gut of flies, from the gut of leeches, to know what kind of animals must be existing. So in Malaysia, they found that there was one animal which they thought had become extinct. When they sequenced one such flesh-eating fly's gut, they actually found that that animal is still there in their forest. There are other interesting things, like you can use the DNA to compare between humans. You can identify DNA from crime scene, sequence them, and then you might be able to find who is the convict. Some very interesting things have happened. I think in my school days, I read this book about Shakespeare's thing called Richard the Third. So Richard the Third had died in a war, and then it was contentious where he was buried. 2012, they excavated his skeleton, and then they could make out from looking at his bone and radioactive dating that this was like 1485, around that time, and it could have belonged to Richard the Third. So there was a living relative whom they tracked out, could be from 19 generation removed. So they sequenced that person's mitochondrial DNA, which does not change so much, and it's like a you can track the maternal lineage, and they could actually show that using this DNA information, they could actually say that the skeleton was Richard the Third. Not only that, then because the DNA sequence tells about predicted eye color, hair color, they could also kind of match it with the portrait, whatever is available about of Richard the Third, and could sort of say that the skeleton definitely belonged to Richard the Third. So you could do such kind of things. You could ask some very interesting questions, that like I mentioned, when did people start wearing clothes? So there's this interesting story that there are these lice which feed on hair, or which feed on the body cloth. So one scientist in Germany thought that if you study the head lice and the body lice, because body lice will be there only if cloth is there. So you could time when the body lice and when the head lice evolved by looking at the DNA sequence, then you might be able to say that when did people start wearing clothes. So what they did was they sequenced the lice specimen from different parts of the world, and they could arrive at that the head lice and the body lice have diverged now nearly 40,000 years back. And then they looked at archaeological information about the needles, and they found that needles would have been there during that time, And then they actually put up this story that maybe humans have started wearing clothes 40,000 years back, because that was the time the body lice evolved from the head lice. So that's the kind of interesting questions. Now you might ask, how is it relevant in today's time? So as you know, coronavirus has put us in the home, and that's why you are listening to all these lectures. What is this coronavirus? How do we know that it has come from bats? How do we know that it is traveling all over the world? How do we track them? How do we track the people who have got the infection? How do we diagnose them fast? So because of genomics, we've been able to sequence a coronavirus genome very rapidly. We've been able to look at the differences, because as it moves from one host to the other, it changes a little bit. So looking at that difference, we can track where it came from. And then also looking at the sequence, we can actually evolve smaller techniques for rapid diagnosis, so that we might be able to know who is positive and who is not. So now it took 13 years to sequence human genome, but as I said, the pace of sequencing has become very fast. So from November or December, when we got to know about the first genome, yesterday in this day, so there's this database, you can go and look at it. There are nearly 10,000 coronavirus genome sequenced from different people from different parts of the world. And people are using these sequences to trace how these viruses would have moved from one part to the other. And so it allows us to look at how the virus is moving, how to trace it, how to track, etc. So this tells us about how having a technology with us, and how having these kinds of toolkits, we can actually address even topical problems like that of today. So these were all about genome sequences. But then the genome is not static, it changes, it differs. So for example, between two humans also we differ at every 1,000 bases. So that way there is no human genome, every genome is different. But then clearly some of them are retained so that we are all human, but some are different because we behave differently, we look different. So now people want to understand how these genomes, even if they have the same kind of framework, what contributes to differences. For example, skin pigmentation, which has evolved to be able to protect ourselves from intensive radiation in the equatorial region. So people are looking at people from different kinds of latitudes, and they actually find that there are certain kinds of genes which are linked to lighter skin color in the Europeans and darker in the Africans, because of which they might have an advantage to survive intense UV radiation. And the same gene are there in the bears, in the zebra fishes, and in these animals, which gives them different kinds of pigmentation. So as I said, having the same letters might also give the same kind of phenotype. So having a knowledge of this, you can actually take the science from different areas, you can actually join them. You can also trace people, human migration. So these colors indicate how close are the DNA to each other. So as you can see, these are the ways why people have traced the route of migration by archaeology. So when DNA was taken of people from all over the world, you could see that there are people who are more similar to each other, then there were differences, then there was some kind of a mixture, then they became similar. And as you overlay these individuals on from where they come from, you can see as you move from Africa to Middle East to Europe to South Central Asia, East Asia, there has been differences in the humans. So the more similar they are, the more are they in one continental population. So now you can actually use this genetic material to trace any new individual and try to see where is their ancestry, where they came from. So we did some such study in India also, because India has been a melting pot for human diversity. Many people, different times of the evolution, there had been settlements, etc. And we at that time it was not known what is the genetic makeup of India. So there was a big project called the Indian Genome Variation Project, and we wanted to know how people of India are related to each other. So this is a language map. And then we took 2,000 people from different parts of the country and tried to ask whether if you reside in the same geography, are you more similar to each other? If you have different language speaking, then are they more similar to each other? So we built something like a genetic map of India. How is that important? So what happens is when you build this kind of a genetic map, then you can start asking questions. For example, this is a malaria map. People reside in very malaria endemic region, and the fact that they reside, they must have some adaptation to be able to survive there. And if you look at their genetic footprint, some genes which are involved in malaria resistance or etc. could be different in people who are residing in different parts of the country. So like this, we get some different stories about overlaying information of people, the climate or the endemicity, and try to see whether we can identify who are people who are at different risks. Why is this important? Because people are wanting to know whether they could use this genetic information to, so all of us are mostly normal. Some might get a disease, and since some if it onsets and it progresses, some might have complications. So the idea is, can you look at the genetic sequence and would know whether how you prevent, how you diagnose, how you know what is the prognosis, how you monitor, how you customize therapy. So basically, how you know whether you can maintain the life and maintain quality of life when people have diseases like diabetes, asthma, etc. So this is what is called as precision medicine, that can you use the genomic information to identify people who are of different risk kind, and can you then tailor the medicine. Now, this is very interesting that 4,000, 5,000 years back, our country had this system of medicine called Ayurveda, where this shloka talks about how Ayurveda deals with maintaining health and maintaining quality of life in people. So we are also doing some interesting things with genomics, where we are trying to see whether we can actually take principles of Ayurveda and use methods of genomic and try to see what is documented in Ayurveda. Can we actually have molecular explanations? That might allow us to find how Ayurveda dealt with lots of diseases, because this system of medicine is still contemporary, and how we can integrate this with modern medicine. So, but for that, we need to provide evidence. So, we have published a lot of papers, and we are trying to create that kind of an evidence. And that is what we call as the science of genomics. Now, just 5 minutes more, where something which might interest you, that why you like different kinds of food. For example, different you go to Subway and you want sandwiches made to be made in a different way, and that is because there are many different components in these food which are perceived by different kinds of taste receptors and olfactory senses. And if you have variations in those taste receptors, then your ability to perceive them becomes different. As a result of which, one might like a burger in one particular way, one like a burger in a different way, and then that would have an intricate connection with how your microbes are liking what you are eating, and that might have a relation to how you behave, etc. So these are the kind of things which also are getting understood, and having this understanding maybe might allow you to have a very good eating behavior also. For example, if you have a bitter taste receptor gene which does not allow you to perceive sugar, then you might or you are super bitter tasters, then you would like these kinds of food, because very little bitter also makes you avoid that food. And so you might be avoiding certain healthy kind of food, and maybe also avoid alcohol and smoking that way. But then if you have too much of these kinds of food, you might have risk of different kinds of diseases. So having known this kind of an information, you might also be better at it. Now people have moved further ahead. They are synthesizing, knowing the genome, that they want to know whether I can synthesize a small virus or a bacteria. This is a lot of medical use. This could be used in different ways, but it could also have a lot of medical use to be able to make a small inactive bacterial genome, etc. Not only to understand biology, but also in various ways. One very last thing I would leave all of you with is that people are using DNA for digital information. For example, we know that there is so many evolution of different kinds of data storage devices, and maybe 2 years back what you use, you cannot use in a different machine. But what is very interesting about DNA is it is 4 chemical letters. You can excavate it from archaeological remains, and the meaning remains the same. So now people are thinking, can they actually convert or information in the digital information 0 1 into AGCT, and synthesize DNA and store it in a small tube, because a very small tube can actually, if you sequence it, it will hold so much of information. So as a result of which, this is very stable, and you can cryptic, meaning make some cryptic information and store it. When you need it, open up the DNA, synthesize it, read it. Now this is like science fiction, but very recently in January, there has been a demonstration that such kind of possibility is there. There are scientists in Switzerland who actually took a small fragment of DNA containing information as to how to print a rabbit in the lab. So they convert that into DNA and put it into beads, and that bead goes into a material which is used for 3D printing. So you print an animal, then you cut out a small piece of that animal, sequence the DNA, and try to see whether from reading the sequence do you get the information back in a digitized way, so that print another animal. So they did this, and you can see taking a small information out of this plastic rabbit, which had its own information about how it is to be printed, they could again print this out. So now this is like the DNA of things. We talk about Internet of Things, but this is the science of tomorrow, because you could then pretty much capture lots of information in a very small space. So I would like to finish now with just a kind of a summary, that once you understand genomes, then you might be able to know various different kinds of things, because it adapts differently, it migrates, there is admixture that creates diversity, not only at the organism level, but at the organismal level. And that can create unique patterns which will tell why biology is different, they look different, or there are different predisposition. And it can actually tell you lots of stories about history, ancestry, culture, climate change, ecosystem, how one thing changes to the other, what you should do to conserve the ecosystem, about origins, about how you are different, applications in forensic, in diagnostics, many things. So I will stop now, and I'm happy to take questions from you. Thank you for patient listening, and I will go back to the camera. Thank you. Am I visible? Hello. Hello.
Hello.
Hello. Yeah.
Your screen is still visible. Can you stop sharing your screen so that we can see you directly?
Okay. I've stopped. Is this fine?
Yes, yes. Now I can see you clearly. So there are a couple of questions which I picked from the chat box. And first
Okay.
of all, thank you for such an interesting talk. Very simple explanation you have given, like genome is a book of life. So I think it is clear to all the students and teachers. So the question which I picked up, a few of them are, um, like how the sequencing could help in the vaccine development for the coronavirus.
Okay. So you need to know. So directly sequencing will not be maybe so informative, but you know which region of the coronavirus is unique and what you need to target. So you can start from that sequence information, but there are many more things which are needed for vaccine development, right? Because you need to identify that what part of virus is foreign and what you need to kill. So there is a lot of things, and immunologists are at work for understanding that. But it has to start with comparing different kinds of coronavirus genomes. You might be able to know what is the unique part of the coronavirus genome which is not there in the other organisms, and that you could target and things like that.
Okay. There is another question related to corona. Like there is a lot of hype in the social media that
Mm-hmm.
this man-made virus. Can sequencing help to identify that it is a man-made or a natural virus?
It is very difficult to say that. I don't, because all of this has evolved. We know that there has been lots of zoonotic transmission from different kinds of animals, because people inhabit are in close proximity, and as our ecosystem is getting disturbed, these animals are also endangered, and lots of viruses then also get transferred during these kinds of events. And it is very so since these have been happening over a period of time, it is very difficult at this point of time to say that whether this genome is synthetic, or whether this is, because this is a very small genome, and it also has shown relation with the other genomes like the SARS and the MERS. So but people are trying to investigate by various kinds of tools, but the data is still contentious. So mostly it looks
Okay.
like all the coronaviruses at least are related to each other. So
Okay. There is one last question related to corona. Is that can coronavirus change the host DNA also?
pardon me, I did not get it. Your voice is
Can
breaking. Your voice is
coronavirus
breaking.
change host DNA?
Can corona?
Yeah. Am I wrong?
Uh,
Virus change host DNA? Host, like whom it is infecting. Can it change its DNA?
can corona change the host DNA? Is that so?
Yes.
Okay.
Yes, yes.
Uh, we do not know because coronavirus genome survives in the host cytoplasm and it replicates over there. If there is a machinery by some way, it signals something to the host nuclear genome and alters the host nuclear genome by event which are called as epigenetic event, then there is a possibility that the host might start evoking different kinds of responses also. So a part of the pathogenesis could be because of the effect of the viruses on the host genome, right? The fact that many of us are falling ill could have to do with our cell machinery is not functioning well, and in some way the virus is harming us, right? So
Okay.
it is possible. Lots of people have shown that epigenetic changes are possible during infection or from viral genomes. So it's quite possible.
Okay, ma'am. Thank you for explaining this. There is one question regarding evolution. Are humans are the most evolved on Earth, or there is another any other evidence for?
So this is what is called as anthropocentric view, that we think that we are very evolved, but if you go to bacterial genome, in its own way, it is very evolved. It is doing in a very evolved manner what it needs to do, right? So it depends on your definition of evolution. Of course, we have got the ability to speak, to think, and to walk, and many other capabilities we have. But evolution is continuing, and it continues in all the life forms of Earth. We take different trajectories to evolution.
Okay. So there is another question is regarding what are the chances of mutation in genome of fossils?
In fossils, so mutations might have happened in the before they became fossilized. So fossils, as since they are not
After
living,
that,
their DNA
mm,
can degrade, and then you might get different kinds of changes, chemical changes in them. But unless we have studied too many fossils, so you will not know whether that predated their fossilization, but unlikely. If there are some microbes
okay.
colonizing on some fossilized things, then that might be changing, but unlikely, at least I do not know. But there are so many surprises.
Okay. Yeah. One another question is I asked by I think some curious student, what if we don't have 23 pairs of chromosome?
What?
Say if we have 20,
So we
if
have
we don't have 23
So
pairs
we
of
have a lot of disease conditions in which, if the chromosome numbers are altered, it leads to lots of different kinds of diseases, like the Down syndrome, the Klinefelter syndrome. So there is an optimum number of chromosomes which are to be there in a genome, and if the numbers, and even if small segments also become duplicated, sometimes it can lead to aberrations in the cellular behavior, and also it could lead to diseases. So every genome has evolved to have an optimum number of chromosomes, but in plants, like in wheat and all, by kind of selective breeding, you can increase the ploidy. In the humans and all, that is not tolerated, but plants, there are many of them which are hexaploid and tetraploid. plants. So it is known in the plant genomes about the ploidy level.
Okay, ma'am. So you also told about Genomics.
Yeah.
So what is the role of genomics in current pandemic?
So, as you might have heard, there are lots of recommendations from Ministry of Ayush also about how they treat fevers. Right? It is possible that what they are seeing as coronavirus, they might not label it as coronavirus, but since these pandemics and since coronavirus have been existing for a long period, millions of years, not this one, but different kinds of coronavirus. So there is a possibility that such kind of fevers would have been seen during the times when Ayurveda was evolving as a science, right? So there would be treatment regimes which people of who avail Ayurveda medicines might be availing. Now, how do people like us who have no knowledge about Ayurveda understand what forms the basis of their treatment, right? How do I understand it in today's language, what they are treating? Many of us might believe it. Many of us want to kind of know for sure how is it working. For example, if you have diabetes, you measure sugar level. So if you have something which you measure, then if you have the medicine, does it go down? So you need to have a molecular evidence, or you need to have something to test it out. So what we are doing in Genomics is testing that, so that what people are recommending, does that have a scientific basis? Many times we might be able to test that out using these modern tools.
Okay, ma'am. Thank you. There is very interesting question from one of the participant. There is a kind of myth or saying that there are seven looking person in the world like you.
Mm-hmm.
So genetic, how much it is true?
So there is a project. So these are called as doppelgangers. I think the term is called doppelgangers, and people have found that in fact there is a community of doppelgangers. That people who look like twins, but they are not. They are from different genetic backgrounds. So maybe by some combinatorial possibilities, their genomes have come to be the same. So people are trying to study them to try to see whether you have the facial features and all. They look similar, and who knows what kind of interesting information, because these are all AGCT. So out of such so many billion people, there might be a combinatorial possibility which would have landed up in the same way. But I really do not know whether it is seven in number. But definitely there is a community of people who tend to come together to say they are doppelgangers from different parts of the world.
Okay, ma'am. Thank you for nicely explaining it. So, a couple of questions have been asked about the PCR method for coronavirus detection. Can you
Yeah.
just put some on this method?
So basically, it is for detection whether it's a viral genome. So you need to have a small sequence which will be allowing you to differentiate between when you have got the infection. So because coronavirus presents as a symptom like flu, right? So flu can be caused by different other kinds of viruses family also. How do you know it is coronavirus? So when you have a genome sequence, then you can actually use these small primer sequences and amplify the genetic material from the virus. Now this is an RNA virus. So first of all, what people do is convert the RNA into cDNA. That is, I showed you translation happens. That is, DNA gets converted to RNA. There is a mechanism which is called as reverse transcription. So you convert the RNA to DNA and make it double stranded. And then you can use these methods called PCR, where you can amplify that. And if you amplify that, then you could, first thing is you can sequence it and match it and compare it, so you will know what it is. But sequencing takes time and it's a little expensive. So you could actually query it by small nucleotides, which will also be able to differentiate it. So many of these technologies are basically being used for doing that. Either you distinguish it by small piece, or you distinguish it by a piece of that DNA, or you sequence the entire genome. So that's how all these PCR based testing, when you hear, or RT-PCR based testing you hear, They are basically trying to distinguish whether the genetic material from the nasal swab of the person belongs to coronavirus.
Okay, ma'am, there is one another very good question. I think after listening your genomics talk, can we increase our immunity by changing our genome?
So you could know what changes your immune system, and you can boost the immunity. So the idea is that all these genes, if they are expressed differently, then you have certain kinds of conditions, and you need to know what has made your immune system go weak, and that is why all maybe different kinds of proteins are being made. And if you know the mechanism by which it becomes weak, then you actually have nutrition or drugs in such a manner that it will start expressing those genes in more manner, so your immune system becomes boosted. So that's the way how you increase muscle mass. So you are basically nourishing your cells with certain kinds of factors which will switch on the genes which build your muscle mass, right? So like that you can actually increase your immune system so that you are able to defend viruses and bacteria in a much better manner.
Okay, ma'am. So we have last couple of questions.
Yeah.
So there is one question: how gene regulation and feedback mechanism works with the changing physiology?
How does can you just repeat it? I didn't, your voice is breaking.
Yeah, how gene regulation
Hmm?
and feedback mechanism
Hmm?
works within physiology.
So that's a very interesting question. So you all realize that as you grow, your body organ sizes increase, right? It does not suddenly increase in your young age. So when you see as you are growing, you might have a small teeth, small hands, or small feet. But as you are growing, all your body is increasing. That means the body somehow, but how does it increase? The cell also needs nourishment, and the cells will divide, and more number of cells increases, as a result of which the body grows. So all these organs have a time a kind of a keeper which tells what is the optimum size. If that is so, then there is some kind of a feedback mechanism which comes from the environment outside and transmits it to the body that now you make something and now it is enough, you stop making that. So that's a feedback mechanism. And when the feedback mechanism actually goes out of sync, for example, you suddenly become fat or your some organs become overgrowth, like in cancer and all, that mechanism of homeostasis is gone, right? So that's a very important part of a homeostatic mechanism where there is a continuous feedback of what goes on from one to the second and how much it is to be made. As it is made, it inhibits the back reaction so that you have a feedback mechanism, and that's how homeostasis is maintained. A break
Okay,
in the
ma'am. So there are last.
homeostasis is the disease state. Yeah.
So there are last two questions. One question is why there is a evolution happening. Why evolution happens,
So that's a
or why
very
humans?
evolution. So a change is a very natural thing, but if the change increases the fitness, then that actually gets selected because it has an advantage, and so every species starts evolving, right? So it is not that evolution is very directed. It is that there would be changes because these changes happen in the DNA at random. But some changes become advantageous. They give more fitness to the next generation. So only if those changes can survive and contribute to fitness, then there is an evolution. So
Okay.
that's Darwin's theory. People are also finding newer ways uh, of addressing newer aspects of evolution, but primarily that has stood the test of time.
Okay, ma'am. So there is one question regarding the recent test which being conducted by CSIR, like even PGI Chandigarh tested that some kind of bacterium. So is it a vaccine or some other kind of medicine? So there is some confusion to the common
No,
people
no, we
about
are
that.
not. CSR is not making any drugs. They're just
There is
testing
some
the.
bacterium strain, ma'am, which is being used as an immune booster.
I am not aware of it. I will not. I will sound ignorant if I tell about it. I am not sure about it.
So I'll share the participant link about it, or maybe we can have one talk on that particular
Sure.
bacterium in
Sure, yeah,
next
sure. You
session.
will have to
So
get somebody from PGI, or I don't know. You have to get that person to
PGI
talk.
is, testing it. Even other companies
PGI
are
is
testing those as a
I I
on
am
testers.
I don't know. I would
Yeah, as per my knowledge, it's not a vaccine. It's basically inactivated bacterium, which is acting as an immune boosters for patients who are
I don't
like
know.
affected
I
with
don't know
corona.
about
So
it.
it helps to.
I'm not sure about it, so I will not want to make a guess.
Okay, ma'am. So, there is last question is like, can coronavirus spread through water? Like, did you do some sampling from water and could see the difference in the sequence?
I did not get it. Your voice is again breaking.
Can coronavirus spread from the water?
Can corona
Like, did you do any samples?
there is
Yeah.
some paper around which has come, I think, yesterday or day before yesterday, where people have actually looked at water around Paris, of whether coronavirus is there. I have yet to read that paper, but I do. Maybe if that comes up, people will have to take it up also.
Okay. Thank you so much, ma'am, for such a nice presentation and answering all the queries. So I would like to inform the participants. So ma'am will be able to reply to your queries even
Yeah,
after this
sure,
talk, because
sure.
this talk is.
I would like to say that there are lots of summer interns in our institute. Our institute, I've not presented any work, but is doing different kinds of work related to sequencing and all. And we have summer interns where, if children are curious, they can come. And then we have a scientist called Dr. Beena Pillai, who runs this outreach programs. So you people are free to approach us, and if you want to understand more or want to spend the summer working with us on some projects, you are all welcome.
Yeah, and before closing, I think one question I think you should answer, which has come
Yeah.
now only. Can you explain the paper strip test developed by IGIB?
So that's a test. Maybe you should actually get Dr. Debojyoti to come, and it's become very popular to present it and let the student hear about it, because I would not like to preempt his lecture. It's very interesting and very different way of detecting, and he's named it Feluda. That's after
Yeah.
Bengali detective, meaning a very popular detective, which is revered by all the Bengalis uh, in the serials and all. So so it is a very specific test which uses a particular kind of a DNA editing technology to detect coronavirus genome, and promises to revolutionize the way the testing is done, because it can be done at the site. So, for example, many we might have problems in remote locations for testing out or reaching out to some central place. So, if when so this technology has been developed in such a manner that it would allow people to kind of test people in remote settings also. But having said that, it's a very interesting technology, because it is also involved in the technology is a part of a bigger aspect of genome editing, which is being used in various kinds of disorders and things like that. And I think you should call Dr. Debujyoti to give a special lecture. He's a wonderful speaker also, and he might be able to tell you better.
Okay, ma'am. Thank you so much. I also request you to answer the questions in the chat box after this talk, because a lot of questions are popping up and it's not possible to answer all here at this platform. So I
So
request you to answer.
how do I do that?
I would let you know after this talk so you can
Okay,
answer those
sure,
questions.
sure,
So
sure.
thank you so much for being with us and spending your time. I also thank all the participants for being with us and listening, talk patiently and putting up a lot of questions. And I also would like to inform that we are having next talk on 1st May related to cancer therapeutics, which will be delivered by Dr. Arvind from IIT Hyderabad. Thank you so much.